Hello and welcome to today's video. Today I will be showing you how BioRankings analytics software can automatically adjust for the batch effect in your untargeted metabolomic data. To begin, I share with you two windows. On my right hand side is the BioRankings analytics homepage. I'm already logged in. However, for you to get here, just go to your browser and type in analytics.biorankings.com. On my left hand side is the metadata file that I have already uploaded to my profile and have analyzed and will be showing you results for shortly. I would like to point out that we work with Washington University School of Medicine in St. Louis, the Gary Patty Lab, to analyze this data, and these results are currently in print and will be published soon. So, in this experiment, we had 324 samples that were tested for COVID. Again, each row represents one of the 324 samples in this metadata file. Column C represents our group column. These 324 samples were given a label of either severe COVID, mild COVID, or they tested negative for COVID. We also found out that these samples were collected in different batches. So we have added column B to our metadata file named batch. And this batch column identifies when samples were collected. So let me explain briefly a couple of samples. We can see that sample in the second row was collected in batch 1 and it had a severe case of COVID. Sample on row 7 was collected in batch 2 and had a severe case of COVID. Sample in row 20 was collected in batch 1, had a severe case of COVID. And by adding the one column that contains the batch information, BioRankings analytics software will automatically adjust for this effect. So let me briefly show you how you would go about analyzing your data that needs a batch effect adjustment. So let's go to the drop-down menu, go to Start Analysis. Again, I've already run this analysis for us, so I will not be running this. But the difference now is that you will select Group Comparison with Batch Adjustment. And now BioRankings analytics software will analyze your data, adjusting for the batch effect. As I mentioned, I've already run this analysis. Here's my result page. I've already loaded our interactive space to review some results. Let's look at some. I currently have highlighted the peak identified at MC 246.07. It was found to be statistically different with a p-value of less than 0.001, and that's actually the adjusted p-value. Our post hoc test shows that the severe samples are different from the negative samples, and our severe samples are different from our mild samples. Let's look to the right hand side of this, our peak plot. Our severe cases of COVID are represented by the green curve. The shaded part represents the 95% confidence intervals for each one of the groups. Our mild cases of COVID are represented in the blue curve, and our negative cases of COVID are represented by the red curve. But we clearly see that our severe cases of COVID have higher intensities than both our negative and mild cases of COVID. This particular feature did not have any candidate metabolite matches when comparing it to HMDB, so we have a blank table. Bottom left corner, we have a box plot representation of our feature. To the right of that, we have our spectrum plot. Again, we have our feature highlighted in yellow that we have currently selected, and next to it we show that there are two other features that have roughly the same RT. Scroll up. look at some different features in this COVID study. Let's look at the first one. Our severe cases in this particular feature are in green. Our mild cases are in blue, and our negative cases are in red. We found after adjustment the p-value was less than 0.001, and our post hoc test found that severe samples were different from both our negative and mild samples, but also our mild samples were different from our negative samples. And in this particular feature, we do have some candidate metabolites we can look at. Again, if you click on the link, that will take you to the HMDB website for that particular metabolite. Let's go back to our results. So with that, one additional column in your metadata file, you can now automatically adjust for the batch effect when using BioRankings analytics software. Thank you very much, and that concludes today's video.